All right, so I'm going to try to do this with the Zoom. I've been using screencasts through QuickTime for the other ones. I thought I would do this a little bit different. We'll see if it loads the Blackboard. So this is the first part of chapter 10, sections 1 through 5, DNA and DNA replication. This would have been a really kind of fun unit to do because we would have built connects models of DNA and worked with the processes that DNA goes through with the connects, but of course we can't meet. So I'm going to do the best I can here. So a little bit about the history. So how do we discover DNA? So in 1940, we knew that there were 4 nitrogenous bases that made up DNA. We knew that proteins were made up of 20 different amino acids and that DNA could possibly be the material responsible for inheritance. There were kind of 2 camps, one for proteins, one for DNA. We're going to kind of go through the experiments that got us to where we are today, which is DNA. So the first thing we have wow, and all of a sudden I have a black screen. Okay, so a different PowerPoint opened up. Let's see if we can go back to the original. There we go. All right, so we have in 1928 during World War I. Most of you may or may not. Maybe I have some history buffs in there. We all know what ended World War II, right? So World War I pretty much ended because the soldiers were all too sick. It was trench warfare. They eat, peed, pooped, slept, everything in their trench. And so there were just a plethora of diseases that were going through the ranks here, and one of which was pneumonia. Now, they knew there were 2 different strains of pneumonia, and what we could do is we would grow them in a petri dish. It's just a dish that has food for the bacteria, and when we looked at them, we could visibly see that they were different. One had what was called a rough texture, and the other one was smooth. And with that, we knew the one that had the rough texture, if we injected it into the mouse, most likely the mouse would live. If we injected it into the mouse, the poor little mouse would die. We know today that what makes the difference between that being rough and smooth is what's called a capsule, and that capsule surrounds the bacteria cell and prevents the immune system from doing its job. Now, they took the virulent strain and they heat killed it, and we know what heat does to proteins. It denatures it. And so when they injected it into the mouse, okay, we know the bacteria basically were kind of killed, and so the mouse lived. So what Griffiths did next was he took the rough living non-virulent strain and mixed it with the smooth heat killed strain, put them together, and the mouse died, which is kind of weird because independently the mice lived, but you put them together, dies. And what he could do was then he would take out the bacteria out of the dead mouse and he would grow it. And what would you when you looked at it, you got smooth bacteria. So it started off as living rough, has now been transformed into smooth. So the question is, well, what did the transforming? What changed it? Was it the DNA? Was it the RNA? So that was the question. So then we have Dr. Avery, who followed up this experiment and said, well, let's break it down into the 4 biological macromolecules, which we studied at the beginning of the semester. So he took the lethal, the smooth pneumonia, he killed them, and then he separated the components out, and he basically did this by adding different enzymes to kill he put lipases to get rid of the lipids, he would put in other proteases to get rid of the proteins, nucleases, get rid of so whatever he wanted, he left that one intact, and he injected it into the mouse with the harmless bacteria. And what we find is that the only mouse that died because the bacteria were transformed is the one that we injected the nucleic acids into. So Avery was like, yep, it's the nucleic acids. But scientists were still like, not enough data, not enough to convince us all. So we still had some people that didn't believe it. So then we have in 1952 what's called the Hershey and Chase experiments. Now with the Hershey and Chase experiments, trying to get my face here out of the way, we have two side-by-side experiments. So what we're doing here is we're using these are called bacterial phages, and bacterial phages are viruses that infect bacteria. And what they do is they put something into the bacterial cell, and the bacterial cell all of a sudden then makes all these viral particles, and then it bursts open the bacterial cell, killing the bacteria, and then releases thousands of viral particles for them to infect more bacterial cells. So the question is, well, what is this virus putting into the cell that's making the cell make more viral particles? So what is transforming literally that bacterial cell into a viral particle factory? What is the instructions? Is it the proteins or is it the nucleic acids? So we do know that we have some differences between proteins, right? Proteins are made up of amino acids. Nucleic acids are made up of nucleotides. Now we know that amino acids have the element sulfur in them. And so we could use a radioactive isotope called S35 to label our proteins. So the first lab, what they did was they labeled all the protein inside the virus with S35. Then they allowed time for the virus to infect the bacterial cell, and then they shook it off. Okay, so they use a blender to shake off the viral particles, and then they use a centrifuge to spin it in a test tube. And so what happened is all the heavier particulates would go to the bottom of the test tube, which is the bacteria, and all the small stuff, the viral particles, would stay in the liquid up top. And then they could see which is radioactive. Was it the liquid with the viral particles, or was it the bacterial cell? And in this case, what we found, and you can see on the left side, the cells were not radioactive. The viral particles were, so the liquid was radioactive, but not the bacterial cells. So that was all at the bottom of the test tube. So they said, okay, let's repeat. So they repeated this with P32, which is phosphorus 32, and we know that we have phosphorus in nucleotides. And so notice how we have the radioactivity inside the head of the virus, and that's where the DNA is kept in the virus, is in the head of this phage. And so they did the same thing. They let the virus infect the bacteria. They shook it off with a blender. They spun it in a centrifuge, and then they checked for radioactivity again, and they found this time it was inside the bacterial cells. So this lab proved without a shadow of a doubt that it was the nucleic acids that were transferred from the virus to the bacteria that gave it the instruction set to make the viral particles. So that is the material that has the instructions on it. That is the transforming factor. So with that information, now we know, oh my goodness, it's DNA. So if we can figure out what DNA is and we can look at its structure, then we could hopefully then figure out what is its function. How does it work? How does it relay that information? How do we go from DNA to a protein? And so this was called the race for the double helix. So we have a lot of different scientists in many different fields that were interested in trying to figure out the structure of DNA. So some of the people that were key players in this were Watson and Crick. Of course, they won the Nobel Prize for this. But in 1953, we have Watson, which is an American postgraduate in England. So when you get your PhD, typically you do what's called a postgrad, which is where it's kind of like a residency, and you get more experience before you become a full professor. And so Watson was very young at the time for his age, and he went to Cambridge, England to do his postdoc. He then was in a conversation with Crick, who Crick was like in his 30s. He was still a graduate student working on his doctorate. They kind of got into a conversation about DNA and found it was fascinating, and they both kind of took that up as a hobby here. And so they started kind of tinkering on this on the side, besides doing the work they were supposed to be doing. Although I really don't know how much work Watson or Crick were doing in their own field. They were kind of really into this DNA puzzle piece here. So another key player is Chargaff. Now Chargaff was a chemist, and what he in New York, and what he was doing was he was taking DNA from different species and analyzing it. And what he found was that the percent of adenine always equaled the percent of thymine, and then the percent of guanine always equaled the percent of cytosine. And so we call these Chargaff's rules. Now, Watson actually attended a dinner party with Chargaff, and that's where he obtained this information. So, this isn't a time where we have internet, so information was not easily available until it was published in a journal, and then you had to wait for that journal to come out. So, what Watson did was when he attended this party, he was like, oh, he took that piece of information with him, put that in his little brain, and took that back to Crick, and they talked about it. And in the meantime, you have in England as well, in King's College, this is all in London, you have Rosalind Franklin, who is an X-ray crystallographer. So that is her profession. Her profession, what she did is she took a material and she crystallized it. She put it into a solid form and then shot X-rays through it and then read their diffraction pattern. So as the X-rays hit that solid, the nucleus of that solid, it would bounce into different directions onto a piece of film, and then you could interpret the structure by its diffraction pattern. So Rosalind Franklin, who, again, not very many women scientists at the time, she was assured by her boss, Maurice Wilkins, that she would have full ownership of this project. She would basically be left alone, and she could do and work, and she would be credited for anything that she came up with. Now, generally, when you are doing research, you do these presentations where you talk about your work in front of your peers. It's like peer review. They'll ask questions, they might make suggestions, and Watson happened to go and attend her peer review session. And so during this, he took a bunch of information, was like, and took that back to Crick again, and then they worked on the model. Now, their first model was completely wrong, and actually Rosalind Franklin and some of the scientists from King's College were invited to look it over for their peer review, and Rosalind Franklin pretty much told them they were idiots and went back to her college. Watson then went back to her and asked for her help, and she kind of looked at them like, what research do you have? Because they weren't doing any research. They were just getting information from other scientists. And she really just wanted to be left alone. She was not in it for winning anything. She just was in it for the science. And she just said, get out. And while Watson was there, he saw that picture again. And when he saw the picture again, he realized where he went wrong. And so he then asked if they could have the model correctly made to the specific measurements they needed, and their bosses, for lack of a better word, said, this is fine, but this is your last chance. And that's all it took, and they were able to take that information and build the structure for DNA. So they did win the Nobel Prize for that particular structure. So it is interesting because they did want to include, although she could have won a Nobel Prize in other fields for her work, but by the time the Nobel Prize was given for this accomplishment, she had died of ovarian cancer. So Maurice Wilkins was added on that Nobel Prize in her place, although he technically did not do anything. So when we look at the structure of DNA, we have our 4 nucleotides. So we've looked at DNA before when we did our biochemistry, our biomacromolecule lab. And so we see we actually have 5 bases, one of which, remember, uracil is in RNA, where thymine is in DNA. So we have our pyrimidines and our purines. Our pyrimidines are our single rings, our purines are our double rings, and then we have our basic structure of our nucleotide, which is, this would be DNA, right? Deoxyribose with our base and our phosphate group. We have our complementary base pairing, adding always base pairs with thymine, guanine with cytosine. So I always remember one pair makes a word, right? So at, that's a word. And then G and C, I always think of the round letters go together. All right. So adenine base pairs with thymine, guanine with cytosine. We have our sugar phosphate backbone. Notice they're going in opposite directions. So this one is going up. This one is going down. Okay. Here is a summary of the DNA replication. So we are going to unzip our DNA, and as we unzip our DNA, we will do so with what's called a helicase, which this helicase here will, I want to give me a pen. Ooh. The helicase here will unwind the DNA, and as it unwinds it, it will also unzip. This is called the replication fork, where our DNA is unzipping. We will have single strand binding proteins that will help prevent the DNA from coming back together. And so as it's opened up, we have DNA polymerase, which, remember, the strands are going in different directions, and we can only add DNA in one direction. And so this is called the leading strand because it's going in the same direction as our helicase. And over here, because again, that strand's going in the opposite direction, it's going this way, that is called the lagging strand. And so what happens is we have to copy it in this direction. So as we basically open up more DNA, we will have a primer go down. That's what gets us started. And then we will have DNA added in this direction. And so it's kind of like, I always refer to it as back stitching. So we kind of back stitch, keep going. And those little fragments that we're backstitching are referred to as Okazaki fragments after the scientists that discovered it. And so what will happen then, the primer will then come out, and we will have another enzyme called DNA ligase that will put those ends together so that our DNA is intact. So there will be several replication forks going on at the same time so that we can replicate fairly fast. If you think about, we were talking about our cell cycle lasting 24 hours, and remember, synthesis is just one part of that cycle. And so it does occur for all the DNA that we have in our cell, it does occur rather fast. So the next thing we would have watched is a crash course, which you could either watch it inside. I don't recommend opening this PowerPoint because it takes forever to download because I've embedded the video, but I do have this video listed below in Blackboard, so in that same folder. And so that is DNA replication in I don't say in a nutshell, but it's pretty much the basics of DNA replication. Please make sure you read this in your books and then watch some of the videos so you have this concept down. All right. I will start the second part. The second part is going to be DNA transcription and translation next. So again, you're going to want to read those together with watching the video.